He's not able to be here in person today, but he was very generous and offered to do a Zoom of his topology and neuroscience tutorial. So take it away, Niko.
Cool. So thanks, Karina, for the invitation. Thank you also to the organizers. Sorry I couldn't be there in person. Some family obligations have kept me in New York City this week. A couple of like logistic things. So first off, can you see my cursor when I move it? Can I point to things?
Yes, we see that.
Okay, awesome. And thank you for using the mic because I noticed during Chad's talk, I cannot hear very well people in the audience without the microphone. So if you have a question, please feel free to interject. I'll try to make to pause often for questions, but I think I'll need the microphone to hear if you have a question. And maybe wave your arms like crazy so I can see in the tiny little screen that I have of the room.
I'll have to run to whoever's asking when bring
Okay.
the mic. We'll try. Thanks.
Okay, thank you. Yeah. And like I said, I'll try to pause often to allow for that. Okay, so this is sort of meant to be a 1-hour crash course in the details of using topological and geometric methods in neuroscience. Chad has already shown you some very cool uses, and this next hour will be a deep dive into some of the things that he maybe swept under the rug for time reasons, and will dovetail nicely because towards the end, I'll sort of wrap up with where he ended as well. Okay, so my goal for the next hour is to answer this question broadly. How do we use topological data analysis in neuroscience? And for many folks in the audience, maybe what is topological data analysis to begin with? And why is it a good tool for this particular context? So in addition to this hour-long talk, I'll also advertise there's a new review article out written by two folks in the audience, Karina Curto and Nikki Sanderson, that has a lot of great answers to this question as well. So let us dive in. If I'm going to tell you about how TDA is used for neuroscience data, let's first just orient ourselves with how neuroscience data generally or often comes to us. So this is a little simulation where over here I'm going to have a mouse running. The red is its head and the blue is its tail. And over here, I'm going to record the activity of one particular neuron. And every time that neuron fires, I'm going to put a little purple dot somewhere on this circle. Where on the circle will be determined by the direction the mouse is currently facing. So whatever direction the mouse is facing when that neuron spikes, I'll put a little purple dot on that sort of compass location on this circle. Obviously, this is a simulation, but the data is real. The underlying data is from multi-silicon probes recording areas in the thalamus and subicular formation of freely moving mice. Hopefully, if I click, this will play. Yep. Okay, so right away, we see some activity concentrated in sort of the westward direction on the circle. And then as the mouse navigates more north and east, we don't see very much activity. But then a little while later, when the mouse sees fit to move back west, now we see even more activity being swept out. Okay, so this neuron seems to really like it when the mouse is heading or looking west, about 180 degrees on the circle. And for a long time, this was sort of the endeavor of neuroscience, right? We would record a particular neuron's activity either as spike trains or more recently with calcium imaging. We would record what the animal is doing, right? Maybe which direction it's facing, what kind of task it's engaged in. And we'd hope that there was some behavioral correlate to the neural activity that would let us determine sort of what the neuron is encoding. In this case, we can draw a nice tuning curve. So we can correlate this particular neuron's activity with the animal's behavior and say, it seems really tuned to head direction. And so on the bottom, I can plot orientation of the mouse, and then I plot firing rate of that neuron as a function of orientation, and I get this nice unimodal peak. Okay, and so this is what we'd call sort of a canonical head direction cell. And this is really cool. I mean, it's amazing that we can do this at all, but there are some limitations to this kind of method. Perhaps the most obvious is simply scale, right? This example I showed you tells us what a single neuron is doing, but you and I have billions of neurons. Even a little fruit fly has about 130,000. So going one neuron at a time is probably going to be a little tedious at best, and maybe we want to think about how populations of neurons encode external variables, not just single neurons. Moreover, in some sense, we got lucky within this example. This neuron happened to have a sort of obvious behavioral correlate. I could pick an x-axis like orientation, and I get this nice unimodal peak. But it is entirely possible that one neuron records or is encoding several external variables, some of which I have access to, some of which I don't. So it's a non-trivial problem to decide what behavior I want to correlate a particular neuron's activity to. Also, there are lots of experimental paradigms where behavioral correlates simply are not available. We might be interested in the activity of these circuits during sleep, right, when the animal is standing still. And maybe even more interesting, during development, before an animal has ever opened its eyes or its ears, what's going on in the brain. There are no behavioral correlates that we have access to. And lastly, this is something that Chad talked about as well. You know, the brain can't compute tuning curves. That would literally be begging the question. It is using the neuron to encode an external variable. It cannot make an XY plot of activity. So this is just all to say that looking at individual tuning curves comes with limitations. One way to get around this is to, rather than recording single units, now with new technology, record entire populations. So this is population activity of some cells in a fruit fly that we'll see later in the presentation. And you can already see that, you know, if I look at the single neuron, there's no obvious pattern, but if I look at the whole population, there seems to be a pretty clear signal here. And so this has become, I would say, maybe the new norm in neuroscience, is record from a whole population. And so this is sort of one of the canonical ways that the data comes to us, is a neuron by time raster. This actual matrix is something I asked someone here at Rockefeller, you know, could you record this? And 3 days later, he gave me an NPZ file that had this matrix. So like, this is a canonical thing we have access to. We can also take this and compute some measure of correlation, similarity across the rows, and compute a symmetric similarity or equivalently dissimilarity matrix. This is sort of the prime object that Chad was discussing. Underlying it is some recording of a time series, generally speaking. I'll pause here to ask if there are any questions before I move on. Okay. So let's, since Chad talked a lot about this side of it, let's consider this sort of data modality and how, you know, what can we do with it? What does this look like? So Chad talked about some visualization techniques. I'm going to go for an even more toy example, just to get a hint at the kinds of things we're looking at. Suppose I've got 3 neurons, and now maybe a mouse instead of a fly. Neuron 1 prefers this heading orientation. Neuron 2, orange, prefers when the mouse is looking at about 180 degrees. Neuron 3 prefers up here. So I'm considering a case of 3 head direction cells that are tiling a circular stimulus space, like so. And maybe let's just record, let's do a toy example where I record the activity of each of these neurons as our mouse forages in an environment. So at time 0, our mouse is looking due west, and so neuron 2 is going to be very active, firing at maybe 50 hertz, some large number, while neurons 1 and 3 are silent, and so I get a vector 0, 50, 0. little bit later, our mouse is now facing sort of southwest, and so I have coactivity of neurons 1 and 2, the blue and orange, neither at their peak, but both active, so maybe both firing at 25 hertz, while neuron 3 still silent. Maybe sometime later, T6, our mouse is not actually upside down, it's just looking sort of east-southeast. Now I have coactivity between neurons 1 and 3, while neuron 2 is relatively silent. And all told, I get a time series or a series of time series. So each row here is the activity of a single neuron through time. And each column is the population activity at a particular point in time. Now I set this up in such a way that I only had 3 neurons. And so this matrix is amenable to just plotting. I could consider each column as a column vector in R3 and plot it on an XYZ axis. And if I do, I get something that looks like this, where we see a circular kind of structure appearing, maybe more of a triangle, but topologically a circle. And so this suggests that I can decode the geometry of what's being encoded using the topology of the thing that gets represented. Okay, so I've told you about head direction cells. This is by no means the only place we find this kind of interesting topology. So in addition to head direction cells, Chad talked quite a bit about simple cells that are tuned to location on retina, but also orientation. And so there's going to be, we expect anyway, a circular feature there. And grid cells exist in entorhinal cortex. So these are a bit more topologically interesting. So if you haven't seen these, I'll just build on what Chad mentioned. So this is a rate map of a particular 2 grid cells in entorhinal cortex, and you see this regular patterning of the environment. And what you can notice is that you can pick 2 basis vectors, I've called them here theta1 and theta2, of the environment. And if I were to walk along, say, theta1, I would encounter this periodic behavior of this neuron. It would fire here. If we're looking at my cursor, it would fire here, then it would go quiet, and then again, and then it would go quiet, and then again, and it would go quiet. And so we could think of this as sort of an R mod Z. It's like the real line, but at every integer, this neuron fires. Topologically, if I take the real numbers and identify all the integers, I get a circle. So I have one circle for this theta1 basis vector. But I also have this other direction in the environment where I have periodic firing, which I've called theta2. And so I really get 2 independent circular coordinate systems that can be wrapped together to form a torus, as Chad showed you a short while ago. So there are lots of neural systems where we see or expect to see interesting topological encodings, circles and tori, for example. Now, it'd be great if we only had 3 neurons every time and we could just draw, in an XYZ plane what was going on. But of course, we generally have dozens or maybe hundreds of neurons encoding a single variable. Again, Chad talked about that with the firing rate diagrams with the little triangular receptive fields. But you get kind of a strange embedding, generally speaking, of what's being encoded. Neurons are also very noisy. And so your signal is going to be corrupted in all sorts of ways. And as I mentioned, these embeddings are generally very nonlinear. So how do we detect these kinds of coding patterns in actual neural data? We're going to use a collection of mathematical tools called persistent homology. And we'll go on sort of a 5 to 10 minute discussion of that now. So the basic fact that I'm hoping you'll take on my word is if I let X be a collection of vertices connected by edges, which maybe form triangles, which maybe form tetrahedra, and I glue them together in suitably appropriate ways, then there is a mathematical construction, a particular vector space called its homology. And it has the property that the dimension of this vector space counts the number of holes in our space. In particular, the nth dimensional homology counts the number of nth dimensional holes in this construction. For now, and really for the rest of the talk, we're just going to focus on n equals 1. So we're going to be looking at circular features in data. So if this is my x, right, I have some vertices and I have some edges. One can ask a computer to compute its homology based only on the information of the vertices and edges, and it will spit out that the dimension of the homology in degree 1 is 1. And not coincidentally, that's counting the fact that there is a hole or there is a cycle in my x here. On the other hand, if I plug this data into the same algorithm and ask it to compute homology, it will say, hey, that the dimension of degree 1 homology is 2. And that is recording the fact that there are 2 independent cycles in this simplicial complex X. One of the advantages of TDA is that we can also, in addition to just considering vertices and edges, we have triangles, we have tetrahedra. Maybe I want to consider these 3 points as neurons. And if they're all really close together in this embedding space, maybe I want to consider them to be one functional unit rather than 3 separate functional units. So maybe I don't want to consider this triangle here actually a whole. It's just one big functional unit. And I can encode that by filling in the hole with a triangle. In this case, taking the perspective that these 3 units are really one functional unit, there's really only one cycle here. And indeed, if I plug this data into my homology algorithm, it will spit out the correct dimension. Any questions on any of that?
We're good, thanks.
Okay, great. Okay, so I've told you now this sort of abstract mathematical, okay, if I have a simplicial complex, I can compute homology. That tells me about shape. But data doesn't come to us like that, right? Data comes to us as discrete data points. So maybe my data looks like this, embedded in some high dimension that I can't have access to, but there's some underlying ground truth that looks like this circle here. But all I have is data points. I have vertices, if anything. How do I construct something useful in such a way that I can detect the fact that there is a big circular feature hiding here? Our intuition is that this is really a circle, and so I want the output of some algorithm to spit out, hey, the dimension of the homology is 1. There is a circular feature here. One of the most naive things we could do is simply to start connecting vertices that are close to each other. And I could turn this bunch of data points maybe into something like a graph. And so I could decide to draw a little radius, a little ball around each of these points at a particular radius, in this case 0.05. And if those balls overlap, I'll connect vertices with an edge. So then I'm left with this, which is not looking much better. I now have both the vertices and a few small edges here. But if I were to plug this data as a simplicial complex into my solver, I would not see a circle. I'd really just see a bunch of discrete points. So maybe our radius just wasn't large enough. Maybe I need to connect more points. So I can draw larger balls around each of these vertices, and I get more edges. With more edges, I now start to see some cycles forming, but those aren't really this global feature that I care about. And these are really close to one another, so maybe I want to consider them to be a single functional unit. And so I'll fill those in with triangle and tetrahedra as appropriate. Okay, so now I've got this, still not a circle. So maybe I just need to go a little bit bigger. And here eventually, okay, at radius 0.25, I have a simplicial complex where there is a cycle. And so if I were to plug this into my homology solver, it would give me the right answer. It would say the dimension of homology is 1, and I would therefore know there is a circle here. But this should bother us for a number of reasons. Even though we have indeed finally seen the right hole, this is not at all useful as a method of discovering topology, right? I had to know which radius to pick, and I had to be able to visualize it. So in the case where data is coming to us, from a neuroscientist, how do I possibly know how to choose the right radius? And the answer is that we don't, or depending on your philosophy, we choose all of them. So I'm going to show you a video now, and what we're going to do is systematically grow those radii, systematically grow the balls around data points, and that'll give me a sequence of these simplicial complexes, one for each choice of radii, and I will compute the homology of each of those simplicial complexes. Whenever a cycle appears or is filled in by triangles and higher dimensional simplices, I'll record that fact over here in what's called a barcode. So I'll play this once with minimal commentary, and then we'll go back and take a closer look at it. Okay, and so here at the end, we start to see one of those long bars that Chad talked about and traced, which is through neural networks. So how did this happen? Okay, so what's going on? Hopefully the beginning is clear enough. I start with my same data points, and I slowly let balls grow, and at each of these radii, I get a particular simplicial complex. Now, right about here, I have a cycle forming here in this pentagon and a little rhombus down here. So I'm going to record the fact that those cycles exist with these two bars here. Okay, the y-axis of the barcode is arbitrary. So these are arbitrarily ordered, but I have two bars forming for each of these two cycles. But pretty soon, these are filled in by my rule of identifying functional units. And so their bars, I'm going to stop recording, right? The bars have died because the holes have filled in. A little while later, I get another little cycle here, but I also see my global circular feature. The little rectangle here corresponds to this bar down here, which is soon to die, right about now. But this global feature, this global cycle, encoded by this bar here, persists for a large range of these radii. And I can keep letting them grow and keep letting them grow and keep adding simplices, but I still have that cycle appearing. Okay, eventually it will get filled in, but now I have this descriptor, this barcode. And importantly, computing this thing on the right did not require any knowledge of this thing on the left. I included it here for visualization, but I can simply take my data, press enter on a keyboard, and out will pop a barcode like this. And I will see, hey, there's a big long bar here, and that's telling me there's a global circular feature to my data. Generally, we regard the small bars as noise, as in this example. I think now is probably a good time to pause for any questions.
Hi.
Hi.
If you increase the radius, you need to have some kind of metric. How do you choose that, or do you just assume the neuroscientists give you the right metric?
Yeah, I would. The latter answer is generally correct. So you can either start with a point cloud, and you embed it in Euclidean space, and then maybe you use the Euclidean metric. We'll actually see a little bit later when using the Euclidean gets you in trouble, and there's reasons to use a different kind of metric, but it's generally context specific. What do you want to consider similar, and what do you want to consider dissimilar?
Okay, thanks.
Yeah.
Hi, so I had a related question. You also seem to have some kind of scale choice here, like what's the scale and maybe the step size. So is that also something that's provided by the context?
So when you say scale, do you mean like the size of these balls?
Yeah, like what's the maximum size you consider, or the step size?
Yeah, so I mean the smallest we consider 0, and the largest would be the maximum dissimilarity. So you go from one extreme where you have just data points to the other extreme of everything's connected. Algorithmically, so you asked about step size as well. Generally, each step is anytime I add something, anytime I add an edge or add an edge or a simplex, I'm going to recompute homology. So the step size is just determined by what gets added to the topological construction.
Thanks.
Thanks. Go ahead.
Great. Yeah. Okay. So why bother doing this? Why? I mean, there's lots of methods for dimensionality reduction, for visualization. Why bother going through all of this mathematics to come up with a barcode? What are the advantages? In the next 20 minutes or so, I'll try to highlight 3 particular advantages. First, it is particularly good at detecting circular, toroidal, and spherical structure, and it is directly quantifiable in a way that many other methods are not. 2, as Chad really beautifully showed, it does not require behavioral correlates. Right? There are learning algorithms, there's CBRA, all sorts of things where I take neural activity and I have some behavioral correlates and I do an embedding based on that. But we've highlighted a number of cases where I don't have behavioral correlates. So TDA is a method that works just as good with or without behavior. And the third, I've sort of titled Functoriality and Beyond, which is simply to say topology is a very old discipline, dating back at least 100 years, depending on how you look at it. And so it comes with this very, very rich toolkit. And so the more we can transform a question, a concrete question in neuroscience to a question in geometry and topology, the more tools we'll have to attack it with. This is not to say these are the only tools or necessarily the best tools, but they are useful tools. And so I'll hint at some of that that also dovetails with what Chad talked about. Okay, so let's focus on advantage number 1 here, detecting and quantifying structure. So this is an example that highlights some of the pitfalls of other dimensionality reduction techniques when you're looking at non-linearly embedded circular topology. So what I'm going to do is take a mouse, record firing rates of some head direction cells. I'm going to treat the activity of these head direction cells as the input to some feed-forward network. I'm not going to tell you what that network is. That's a whole separate story. But suffice to say, I'm going to sort of screw around with the embedding, but hopefully preserve the information. And then I'll look at the activity I get downstream. And I might be interested to know, is the information initially encoded by my head direction cells preserved through this network, i.e., does it show up downstream? So what I'll show you now is the output of looking at these downstream neurons, both through the lens of UMAP, projecting on the original data, and computing barcodes of the original data for 3 different architectures. So in the first architecture, UMAP gives me this, which is not super promising. In a different network, I get this thing, which looks pretty circular. And in network 3, I get like something that definitely must be a circle, or so we think. But here's the thing. UMAP is taking, in this case, about a 100-dimensional object and shoving it down into 2 dimensions. I also have to pick parameters for this. I have to pick in particular, the number of neighbors, and this can in the algorithm, and this can drastically affect the output I get. So there's choice involved in this UMAP projection, and it's some algorithm that's shoving 100 dimensions down into 2. On the other hand, it'd be nice if I had a method that worked directly with the data of these L3 neurons and didn't, just treated them as it were and didn't shove 100 dimensions down into 2. And if I do that, or the barcode is one way to do that, in this first network, I see a significant feature. So the data there is circular structure in the data. It's just not being found by UMAP. So that network 1, we could say, is sort of a UMAP false negative. Network 2, the two methods agree. UMAP sees a circle, my barcode sees a circle. But network 3 is sort of a false positive for UMAP. It thinks there's a circle, but if I look at the data directly, I really don't see that. Another thing to highlight here is these barcodes and corresponding persistence diagrams that Chad talked about come with a whole host of statistical tools. There are ways to quantify when a bar is big enough or significant, and there are ways to compare persistence diagrams across data sets that something like UMAP just doesn't come with. So this example is meant to highlight topology is particularly good at finding circular features in data and quantifying them. We could also ask about more complex topology. So Chad mentioned these grid cells, and we talked about them a little while ago, that we think grid cells, their population activity lives on a torus. This was shown very beautifully in a recent paper from the Moser lab and collaboration with some topologists. So these are the rate maps for sensible grid cells. And these are the persistence diagrams they get, the barcodes they get. And we see in each case, this is rat1, rat2, and rat3, 2 very long bars in dimension 1 homology. And they do some various statistical tests to sort of quantify their significance. And all in all, these barcodes highlight that there is a toroidal manifold underlying the encoding of grid cells. So you look at this, you look at the barcodes, and it's plain to see that there's a torus. This up here is what UMAP spits out. Arguably a torus, but they had to very carefully select parameters to get this torus to show up. And if you had to select parameters to get it to show up and it doesn't show up with other parameters and you can't quantify it, I would argue it's not the best method for direct analysis of data. It can be informative, but it's hard to do rigorous quantitative studies with that sort of thing. But you can with barcodes. Okay. Oh yeah, and then things get even worse if you've got conjunctively tuned grid cells. So these are cells that are tuned to the grid and so participate in that torus, but are also tuned to head direction. And so from those, we might expect to see 3 circular features, which we do in the barcode over here, but which is completely opaque both to Zebra, a new neural network embedding, and UMAP. Okay. So particularly good for these kinds of encodings. Okay, that was it for advantage 1. So I'll pause here if there are any questions.
That's fine. Go ahead.
Okay. Okay, so advantage number 2, I can do all of this analysis without having any clear behavioral correlates. To talk about this, I'm going to introduce one other tool, which is called a Betti curve. I build a Betti curve from a barcode. It uses only the information from the barcode. What I'm going to do is simply, for each dimension, so let's focus on H0 here. At each point on the x-axis, I'll just count how many bars are alive. So here at time 1, there are, I guess, 14 blue bars alive. So my blue curve starts at 14. A while later, here, some of these bars die, and so there's only 10 blue bars alive. So my blue curve drops to 10. so on and so forth for each dimension in the barcode. Okay, so from a barcode, I can build a Betti curve, and that's what this guy is. One of the first papers exploring the uses of TDA in neuroscience was from Trina and Chad and Vladimir and some experimental collaborators, where they looked at the Betti curves of a canonical complex associated to a dissimilarity matrix that came from place cells. So they recorded ostensible place cells in hippocampus and looked at the Betti curves that they got. As a null model, one can start with a totally random matrix. And the Betti curves one gets from this construction are very stereotyped. And they look like this. So this is in dimensions 1, 2, and 3. Pay attention, the y-axis is quite large here. And the Betti curves of these random complexes follow a particular pattern where the peak increases with dimension. So the peak of Betti 2 here is larger than the peak of Betti 1. Peak of Betti 3 larger than the peak of Betti 2. Okay, so this is just a null model for these Betti curves. On the other hand, if we build the complex associated where sort of the underlying data is randomly sampled points in Euclidean space, we see the opposite trend. That is, as I increase the homology dimension, 1, 2, 3, the peaks of the Betti curves go down. And you'll notice the y-axis here is quite a bit smaller than the y-axis previously. So the hypothesis was that since place cells are geometrically correlated, right, based on the overlap of their receptive fields in the environment, the Betti curves one would expect to obtain from that neural data would follow something that looked closer to the Euclidean distribution. And indeed, that is what they find. So these are the Betti curves from that place cell data, and you'll notice y-axis only goes up to about 40 here, and you see this decreasing trend as I increase the dimension of Betti. Okay, and so this is the data versus those geometric null models, or geometric models, and the data versus that completely random null model. Okay, so qualitatively very different. And again, these come with nice statistics. One, obvious thing we can do is simply compute the area under these curves. And you see the area under the curve here for G is for geometric, stays quite low, whereas their shuffled area under curve is rapidly increasing. Okay, so this method detects the difference between G's geometrically correlated neurons and just sort of random correlations. Importantly, they didn't take this as their only null model. You can also compare to the sorts of distributions you see if you take the place field and just scramble it. Okay, so this is sort of a more faithful null model. And so I've kept all the information about the cell, like its average firing rate, so on and so forth. I've just destroyed the geometry. And again, the Betti curves detect that difference. So G is for the geometric, that's their raw data. And then you see the scrambled place fields are quite a bit different, and the shuffled is more different still. So they're able to detect this kind of correlation structure from the neural data alone without any reference to behavioral correlates. And they can do so quantitatively with lots of nice statistics. And very coolly, they see these same kind of differences during non-spatial behavior. So during wheel running, when the animal sort of maybe thinks it's moving but isn't, and during sleep. And so these are explicit cases where you can't compute rate maps or tuning curves, but nevertheless, that correlation structure persists and is uncovered by these methods. Okay, one more example of this, these cases where I don't have behavioral correlates. So we'll go back to the barcodes. So I showed you earlier these barcodes for grid cells during basically awake exploration. So these are 3 rats during open field. OF is for open field exploration. We see the hallmark barcodes of a torus here, as well as during wagon wheel. That's this sort of wagon wheel looking maze here. But remarkably, the same type of population activity persists during REM sleep and slow wave sleep. And that was actually one of the key questions this paper was asking in the first place is, are grid cells actually well modeled by a continuous attractor network? If so, their activity should persist across states. And so here's a case where you really needed topology to answer the question. I really needed to understand the correlation structure of my neurons without behavioral correlates to understand what kind of model, or what kind of encoding is being used. And coming soon, I've been told sometime this summer, although I guess it's almost fall, so sometime this fall, they study the same kind of thing of the same grid cell system during development. And they see the emergence of the grid system before the animal has ever opened its eyes or ears or done any kind of exploration. So, and it's a very cool, very clear, like on day 10 or something, you go from no grid to grid cell online. And that's another case where, there's no hope of a behavioral correlate, but by looking at the data alone using these topological tools, you uncover something, a very cool mechanism about the formation of the grid cell system. Okay, that's it for advantage 2. So again, I'll pause for questions. I think I can see. So if someone raises their hand, I'll be able to see. And if not, I'll know I can move on. And
Any
looks like I can.
questions?
Cool.
I guess go ahead.
Okay, great. So advantage 3, looks like I got 15 minutes. Is that correct? 10 minutes?
Yeah, that's right, including questions at the end.
Okay, great. So this is a bit shorter, and I want to show you some real data. So I'll maybe go through this a bit quicker, because this is something Chad talked about. So we've, thus far I've talked about all of these systems sort of in isolation, and detecting structure in a single neural population. But of course, the brain passes information, like crazy. When I look at my dog, something happens in V1, but that gets sent to V2 and other higher areas. And so if maybe I replace my dog with a Gabor filter, right, I expect an S1 in the data to induce that sort of encoding in V1. Even more complex, we know there are head direction cells in the thalamus and subicular formation, but near the hippocampus. And I know there are head direction and grid conjunctively tuned cells in entorhinal cortex. And moreover, that hippocampus and entorhinal cortex talk to each other constantly. And so this suggests that, these kinds of encodings are being passed and propagated through networks. How can I hope to track them? Well, that's what Chad told you about. Right, so they do this great simulation work studying how topological structure moves across neural populations, and they explicitly do that example I showed you where you're looking at a mouse viewing a Gabor filter, and they see circular topology in V1, and they match that to circular topology in the anterolateral region. So since Chad talked about this, I guess I won't go into it much further, other than to say, this is using like a very cool feature of homology, namely that it's functorial, to answer a real question in neuroscience. And functoriality is one of many tools that exist for these kind of topological invariants. And so I would suggest there are just bundles and bundles left to explore, and that can be useful for neuroscientific data. Okay, and they do grid cells as well. But okay, since I'm running out of time, let's do some of this on actual data. So I made a little repo here. If anyone wants to follow along, it is publicly available, and there are lots of other examples. I'll just go through one where we're going to get our hands dirty and use TDA to find a circle in some actual neural data. The neural data that we're going to look at comes to us from the fruit fly. So this is a 2-minute crash course in the fruit fly anatomy. They have this handlebar-looking group of neurons called the protocerebral bridge. And there are these specific cells called EPG cells. The cell body is somewhere up here, but it projects to both the protocerebral bridge and this ellipsoid body, which is a literal ring of neurons, physically a ring of neurons in the fruit fly brain. And it's been beautifully shown that this collection of neurons is responsible for tracking the heading of the fruit fly. How do we know this? I'll show you a video. So what they've got is a fruit fly here, suspended on a little stick, lowered onto an air suspended sphere, a little ball. We're going to see the fruit fly moving, and then they have this closed loop system where if the fly moves, let's see if I can get this right. If the fly moves clockwise, this bar on this virtual reality screen will move counterclockwise. And so it's acting as the fly's virtual sun. So this is all an elaborate experimental setup to make the fly think it is actually walking and has a visual cue when it changes direction. And then up here, we're going to see the activity of neurons in that protocerebral bridge. And we're going to see 2 bumps, sometimes 3, moving along the bridge in coordination with the direction the fly is walking. It's calcium imaging, so there's some latency, but you can hopefully see this bump on the bridge moving with the bar. A little while later, I know the fly does a big movement. Yeah, see there, and so you get this bump of activity moving rapidly across the bridge. Okay, so there ought to be a circle here. I'll show you today some data collected by Stephen Thornquist, a postdoc in Gabby Maimon's lab here at Rockefeller. Just to give you an idea of the state of the art, like I asked Stephen, like, could you do this? And 2 days later, he was like, oh yeah, sure, here you go. Like they're very good at this. It's very cool. So let's take a look at the data. Am I still screen sharing, or has that disappeared?
Yes, we see your screen.
Okay,
It's not full
hopefully
screen, but okay, now
is it a VS
credit
Code?
imports and functions.
Yeah, okay, perfect. Okay. So I'll try to wrap up here quickly, but I want to show you at least one example. So again, this is publicly available if you'd like to download or look at it later. There are just some helper functions, blah, 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 blah. So here's the data. I got this on a flash drive. Like he gave me an NPZ file. Like this is actually how data can come to us. So the keys are as follows. What we care about is this df of f. So this is the change in fluorescence of that calcium image over the baseline. So I'll just extract that and I'll plot it. And there's my data. So this is 16 neurons across the bridge, about 20,000 data points. The heat map is just this fluorescence, raw fluorescence. You'll notice that it gets quite a bit brighter here, right around the, I don't know, 6,000 mark. This is when you turn that blue bar on. So before, the fly was just wandering in darkness, and then those cells really activated when the bar is turned on. Okay, we can extract some information. Let's just take as our data the robust activity when the sun is on. So I've got about 12,000 data points. I'm treating each column as a data point in 16-dimensional space. I'll downsample this for the sake of my computer, and I will do persistent homology on this point cloud. Now, I've shown you, like, there has got to be a circle here, right? I mean, it's tracking the heading direction. These are compass neurons. Like, if ever there was a time to use topology, it would probably be now. I've computed the barcode. Let's plot it. And maybe there's an obvious bar here, but that one's also pretty long, and so crap. So what happened? Why didn't this work? This is also meant as an introduction to the real challenges of doing TDA with neuroscience data. As it turns out, these cells are tuned not only to head direction, but also to speed and angular velocity. So a particular cell might be encoding the north direction, but its activity is also modulated by how fast the fly is walking and the angular velocity with which it passed through the north. And so rather than getting a nice clean circle, you end up with sort of a really smeared annulus that's maybe smeared all the way down to the origin. And so I don't see the circle there. But not all hope is lost. We can account for that, and Stephen realized this. So if we simply take our knowledge that, hey, these cells are also tuned to maybe speed, we can normalize our data and then use cosine distance instead of Euclidean distance. And now we see in that same barcode a very obvious feature. If you are worried that this feature is artificial, that I massaged my data just so I saw it, we can also make sure we didn't. So I can shuffle all of the same data and use that same normalization and cosine distance pipeline. And we expect to see no structure now that I've destroyed it. And oh, my computer is frozen. Oh no. Oh gosh. Hopefully you'll take my word for it since I'm running out of time, or I could maybe I'll try this. See how quickly this will go. Okay, here we go. So, okay, no bars. So when I destroy the structure, no bars. We did not cheat. Okay, I'm right about at time. So I think with that, I will just thank you all for your attention, and thanks again to the organizers.
Thanks, Niko. Questions?
Yes, thank you very much for this. It's really interesting. I'm wondering, so now this is a kind of very special setting, right? So you know there's some kind of circular feature, and you try to find that circular feature. So can you comment on in which cases you can use that? So can you also detect like more complex features, and which paradigms do you, or yeah, so to which kind of paradigms can you apply that?
Yeah, great question. So I mean, you're absolutely right. In this case, I can literally image these neurons and I can do, behavioral correlates. I would say, regarding this example, one interesting paradigm might be, to look at these cells during sleep or during different activities. But more generally, okay, I've shown you circles, I've shown you tori. We know these tools can detect spheres, and I think, I don't know, I and many others hope to find a spherical stimulus space using these tools. But I think maybe the most direct answer to your question, like if you have a particular encoding you're looking for, a circle or a torus, these tools are good. But Betti curves are, in some sense, let's say, can be better for exploration. So just looking at long bars tells me about topology, but Betti curves detect geometry as well. So if I were to sample points from a circle and a cylinder, topologically they're the same, and I'd see one long bar in the barcode, but they would have different noise profiles and can be differentiated by that using Betti curves. So there are all sorts of other kinds of stimulus spaces that we might be able to detect using something like Betti curves, rather than just looking at long bars in a barcode. Is that what you were asking, or did I not quite?
Yes, yes. But so the dimensionality should be quite low, right? Or does it also work for high dimensional features?
You mean sort of like the latent dimension, like if I've got 8 circles in my encoding?
No, but if I'm not interested in circles, but spheres or like higher, I mean, I can also increase the dimensionality there.
Yeah. So certainly we can detect spheres, and I mean, in theory, we can compute homology in any dimension, and that'll detect, I'll go from detecting circles to detecting voids to detecting the higher dimensional analogs thereof. And computationally, things get a little dicey, but one can do it.
So what's the highest which is computationally feasible, the highest dimension?
I think feasibly probably 3 currently. Others in the room can correct me, but I've only ever seen it computed up to dimension 3.
Thanks.
Thank you for your talk. It's very interesting for me. But I'm now like in this field. Mostly my research experience the neural mass model. So we usually have a 2 network structural connectivity or the functional connectivity. Can we using your tool to calculate the like a barcodes for different network, then to see how those the topology is different from different network? Also that like that's could be like a resting state. If you have like a external input, those to change the network, can you just using your to like recover actually the what part of like structural connectivity changing to make this functional connectivity? You'll see the signal propagation. Thank you.
Yeah, fantastic question. Yeah, so let me take that maybe in 2 parts. So I think first you asked about like structural connectivity, maybe anatomical connectivity, and that's been used widely. It's something I didn't mention here only for time reasons, but applying these tools to connectomes has been very, very effective. And I would point you to this article, which beautifully talks about some of the applications where I take, rather than just like a directed graph, I could start considering triangles and tetrahedra and so forth. And then your second question, I think, was about functional connectivity in different networks. That's actually something I'm actively working on, and other folks are too. So absolutely, we hope to use these tools to uncover mechanism from observation of activity. I don't have a concrete answer for you yet, other than to say that's an active area of research.
Others? Okay, well, let's thank the speaker again.
Thank you.
So we'll break for lunch and see everybody back at around 1:30.